It's the second dimension. First dimension is isoelectric focusing. Second dimension is what we did in the 1D. We separate by their molecular weight, their mass, okay, with SDS. So therefore, in this way, complex mixtures of thousands of proteins can be resolved. So this is kind of I've got two overview slides. This is slide number one. And this is called IEF. This is the focusing strip. And those little blots are bands of proteins. Those are not single proteins. Those are mixtures of proteins that have the same isoelectric point, their same PI. So you do this kind of focusing, and then you take the strip, put it and load it. Instead of the wells, you actually put it in on top of a slab gel and apply current, and proteins then move from each band according to their size. Here's a protein, here's a protein. So that's said to be the second dimension. So if you find an interesting spot, a spot is a protein. Imagine two different cell types, cancer, non-cancer. One has a spot, one doesn't. You could actually do a peptide fragment digestion and look at the fingerprint and try to identify it, or we can do Edman degradation, K-E-D, Edman degradation. Okay, and we'll talk about that. And this is, we've kind of talked about the mass spec charge to mass ratio and get a fingerprint. But anyway, there the spot is. And this is showing the different conditions. So condition one, condition two, and we compare those. So we solubilize, we do isoelectric focusing in the first dimension. And then we apply and put these strips or capillary little tubes, get the gel out and lay them in these big slabs. And then we apply current. And then we stain. We can stain the proteins with Kamasi. Cheap, but not very sensitive. Silver, expensive, but very sensitive. Fluorescence, or we radio labeled these proteins with S35 methionine. Almost every protein has methionine amino acids, so that's sulfur 35, that's an isotope. And somehow we're using silver Kamasi fluorescent staining or audio radiography, where we're detecting by X-ray film proteins that have S35. Either way, you have situations where you're looking at the two. We have software that can scan kind of like a bingo card and know the exact coordinates, kind of like a map. And then we can compare the two conditions, and they're showing by arrows two spots that are in B that are not in A. You can excise those spots and apply a detection method such as a mass spec or Edman degradation, whatever at that point. You can take those spots and make antibodies as well. So there's multiple things to do after you've determined interesting spots. So this whole process, you may have heard the term proteomics. This is where we're looking top down at everything and trying to spot differences. So this was a big movement. Again, when science was starting out as far as looking at proteins, we were stuck kind of at the bottom, looking at proteins one at a time. Here we're looking at all the proteins across the landscape and seeing what's different, which proteins are high expressed, low expressed under the conditions, and I'm not defining the conditions. It could be diseases or other kinds of conditions. So this isoelectric focusing is you apply a sample to the strips or the gel, and you have low pH or high pH on either side, and you apply electrical current, and proteins will move until the movement between the two poles is equal, and that's said to be their pi. Okay, they will now focus right there. Focus means they don't move. And so here in this cartoon, there's 4 different proteins in this mix, and we separate them that way. So here's another figure. High pH, low pH, we apply the sample, say, to the low pH, and they will move. The ones that are basic proteins will move towards the low pH, so they have a negative charge. Proteins that have a net positive charge will tend to stay here, right? Those are also known as acidic proteins with a positive charge, and negative or base proteins with a negative. And so here we are, we apply this to an SDS page, the second dimension, and separate. So notice this right here, this yellow band. It's not one protein, it's 2. One's big, one's small, okay? And so this one here shows a very large protein and a small protein. So you can see the idea now. We're separating one dimension and then separating in the other. And so you end up getting spots that can be further analyzed. So here's isoelectric focusing, looking at these 5 different proteins in color, and you apply an electrical force across a pH gradient. The strips have a pH gradient from high pH, pH 14, to low, from very acidic to very basic. At low pH, most proteins have a positive, while at high, most proteins have a negative. So at the extremes, these are positively charged, this is negative. And so these are going to come this way, those proteins are going to come that way. And at some point, the proteins will arrive at a destination on the strip where they will stop. In other words, the forces from this pole and that pole are the same, and that occurs at a pH level is unique for that protein, depending on the R groups, okay, and they've reached their isoelectric point or focusing. They stop. So now we've separated these proteins by their R group charge. Okay, that's so the cartoon here just shows a mixed sample, and of course each protein group, and these are different proteins necessarily, end up focused where their isoelectric focusing point is, and that is determined by the R groups that they have. Okay, so different R groups. So that's the first dimension. And then you, if it's a little capillary tube, you pop the gel out, and there's some agarose you can put right there, and then put it in a tank and apply electrical current to here, and separated by the second dimension. So first dimension, second dimension. And here are the spots here that you can visualize by staining. So remember, we're going back to preparing our sample. Okay, so we may have popped the cells open, all the techniques we talked about. We could separate the cell by the, the facts. Okay, so try to put these techniques together here, facts, and we get certain types of cells, and then we can break them open and get the different organelles, and then we could take those fractions and apply two-dimensional electrophoresis here. And it talks about the level of detection, about 100 nanogram or 2 picomoles in 1 liter of culture. So this is medium abundant proteins, and you can do all sorts of different purifications, extractions, get rid of the DNA, RNA. Here's the subcellular fractionation by the different centrifugations, the different chromatographies we've talked about. If you have an antibody, you can precipitate, or other kinds of affinity pure selections here. So all the methods that we had talked about can slide in, and then you can use this 2D gel electrophoresis as a way to get, again, the spots. And we're talking focusing, just showing the cartoon here, the different focusing, and we get the focus proteins lined up, and see the size difference here. It's like the Earth and the Moon kind of here, and so those line up, and then you apply here. So it's a good resolution method, and each protein can be measured, and now they have software that can predict. So bioinformatics software, so any protein, and you may have done that in your reports, you can predict the isoelectric focus, the PI of proteins, and determine the difference there. So some companies, instead of capillary tubes, actually have pH gradient strips. They call them IPGs, and they come in a little container. And these strips have different pH ranges. So here's the broadest from 3 to 10, but you can do 5 to 8. So if you have an idea of the nature of your proteins, you might be able to focus in more finely with a focus strip. But anyway, you got different strips, and you can apply a sample. And here it is. Here's the container it loads in, the strip, and here's the sample being applied. It's hard to tell, but this is the sample chamber. Here's another company sample chamber. And then they apply the electric current and then generate the isoelectric focusing here. And then they apply it to a slab gel, and you get the spot separation here. And again, this just shows when you isoelectric, you see it's showing the velocity here of the proteins. Here's the big protein and a small one here separated there. And then different instruments that are available, so you can scale up with the size if you need. If there's a lot of proteins, you can, the bigger the slab separation, the more you can fine tune it. It depends on what you're doing. Here's some more instrumentation here. Just wanted you to kind of see. And so once you get the spots, you have to figure out a way to visualize it. You can stain it with Kamasi or a silver stain. You can do some type of immunodetection after you blot it, and we'll talk about blotting. Or you can radio label proteins with S35 methionine and visualize with X-ray film. So either way, you've actually got a gel and there are spots, and the imager can actually see those spots and record the actual results. So think about all the diseases and variants. Talking about personalized medicine, we could look at your spots, and then in your checkup, just look at your spots every year. Hey, you got something weird happening this year. This new spot has gone down or, souped up. So this is part of the big proteomics, big landscaping view here that we can apply to diseases and people situations. Here is a healthy control individual. Here's some type of disease, and they've shown you in the box. Take a look at this. This spot is there. It is not there here. So apparently in healthy, you make this protein, but if you have this disease, it doesn't say what disease it is, then the spot is gone. So is not having that protein, does that correlate to the disease? Okay. What is that protein? Well, we can do a trypsin digestion, do a protein fingerprint, or we can do Edmond degradation. Okay, Edmond degradation, and we'll talk about that. So here's just the isoelectric focus here. I just found another picture, and it's pretty blue, showing the gradient. See the gradient concept from positive to negative charges here. And then here's basic amino acids, peptides, basic proteins, acidic base proteins. And here's the separation. So these are high molecular weight acidic, basic, high molecular weight acidic, low molecular weight basic, low molecular weight acidic. So you can, and these proteins would kind of be neutral here, right? So high, medium, and low. So now you can have the imager actually kind of like a bingo card, obviously more than a bingo card, but each spot has its own place. And so you can look at all sorts of different diseases, situations, drug treatments, and just look at the grand scheme of the protein production in those cells. And then you can figure out, some proteins that we should start studying. Okay. And we're going to talk a little bit about Western blotting, how we actually apply all these proteins to a piece of nitrocellulose or PVD nylon membrane, and then apply an antibody and visualize proteins that are specific to certain antibodies. So it helps you identify what those proteins are if an antibody recognizes them here. So here's the first dimension, second dimension, and the different ranges. You can get a broad range, you can get a little bit narrow range in pHs here. Different size gels to separate out the second dimension a little bit. Then we can stain. And if you stain, you can still digest each spot. So you can actually go in and pick by scalpel or some other type of method. They actually have robots. You stick this on and they'll go to an area and go through the acrylamide gel and pick that spot that then can be digested, and then you can look at the fingerprint. We can do Edmond degradation, and that's the sequencing. Or you can do silver stain. This is Kamasi, but silver staining as well. So this is not the end. This is just the beginning of the beginning, right? Or the end of the beginning. And now we're ready to figure out, well, what's the difference between these two types of cells or two types of organelles? What does the ER and Golgi look like? What's the Golgi look like or the mitochondria look like? So if you had a protein that sometimes went into the nucleus and sometimes in the mitochondria, this kind of analysis would highlight that protein and say, hey, sometimes it's here, sometimes it's there, especially if I had an antibody against it. I don't know, if I had an inquiry, maybe that's some of this stuff might apply to that. So that's my 10-cent tour of two-dimensional gel electrophoresis. We're taking situations, cell types, organelles, samples, and comparing them looking at a grand scale of all the proteins in those cells or samples or conditions. And then we're setting up to identify the differences there, what's missing, what's new, what's lost, and try to correlate that to some clinical aspect of humans. So we'll leave you with that, and we'll pick up with the next topic, which will be getting into Edmond degradation and mass spec. Come back. We'll have fun. Yay.